morning and welcome everyone to Ahmad Dahlan International Conference on Health Sciences 2025. On behalf of Faculty of Pharmacy, Public Health and Medicine of Universitas Ahmad Dahlan, I would like to extend a warm welcome to all of you to taking for the time and attend this event. It gives me really great pleasure to be the anchor for this conference, and I'm really excited to be hosting it. I would like to take a moment to express our immense gratitude to Faculty of Pharmacy, Faculty of Public Health, Faculty of Medicine, and of Universitas Ahmad Dahlan who have made this conference possible. Because we know that without their support, we would not be here today. And we are really, truly thankful for their efforts and contributions toward this event today. Ladies and gentlemen, before we begin, I would like to take a few moments to explain the rules for today's conference. First, please make sure your Zoom meetings are on mute so we don't disturb the speakers and the other attendees. And the second of all, we encourage you to all participate in the discussion and ask the question related to the conference topic for today. And the Q&A session at the end of the session with our moderators for today. And please make sure you keep on your question brief and to the point to allow for a productive discussion. And the third, please make sure you stay for the duration for the conference as it will provide a holistic understanding of the topic. We hope that you will adhere to these rules and participate fully in the conference, and thank you for your cooperation. Without further ado, let's begin the conference. Bismillahirrahmanirrahim. Distinguished guests, keynote speakers, presenters, and all participants, Assalamualaikum warahmatullahi wabarakatuh. Assalatu wassalamu ala asrofil anbiya'i wal mursalin, Sayyidina wa maulana Muhammadin wa ala alihi wa sahbihi ajmain amma ba'du. Good morning, ladies and gentlemen. My name is Putri Rahma Novitasari. In this wonderful and precious chance for me to be your master of ceremony for today, in 20th of May 2025. We have an exciting panel of speakers lined up for you today, and we hope that you will find their presentations and discussions informative and engaging. It is a great honor for us to welcome you to the opening ceremony of Ahmad Dahlan International Conference on Health Sciences 2025. This year's theme is One Health, an integrated approach to sustainably balance and optimize the health people, animals, and ecosystem. First of all, let's say thanks to Allah Subhanahu wa Ta'ala who has been giving us guidance, happiness, healthy, and mercy so we can attend this, participate, and this event today without any obstacles. Praise and salutation upon our Prophet Muhammad Sallallahu Alaihi Wasallam, the last messenger and the best figure in the universe. In this beautiful moment, I would like to say welcome to Dr. Apotheker Iis Wahyuningsih Magister of Science as a Dean of Faculty of Pharmacy Universitas Ahmad Dahlan, Ms. Roshidah Philosophy of Doctor as a Dean of Faculty of Public Health Universitas Ahmad Dahlan, and also Professor Dr. Rusdi Lamsudin, Specialist for Saraf Medical Science as a Dean of Faculty of Medicine Universitas Ahmad Dahlan, and the other Honorable Vice Dean and Head of Study Programs of Faculty of Pharmacy, Public Health, and Medicine of Universitas Ahmad Dahlan, and also distinguished lecturers, and of course, all of my beloved participants. Well, ladies and gentlemen, on this special occasion, I would like to say you about the several agendas for today. So allow me to read several sequences of our agendas. The first is the opening ceremony, and the second is reciting Holy Quran, And the third is singing national anthem of Indonesia Raya and Marsang Muhammadiyah. And then the fourth is welcome speeches by Chairman of Ahmad Dahlan International Conference of Health Sciences 2025, and the Dean of Faculty of Pharmacy Universitas Ahmad Dahlan. And the fifth is the speeches by keynote speaker Professor Takahashi Yashiro, Philosophy of Doctor from Jichi Medical University of Japan, and Associate Professor İlker Ateş from Ankara University of Turkey. The sixth is the questions and answer session, and then the seventh is lunch break, and the last is the parallel session. Well, ladies and gentlemen, so I would like to start this session by reciting Basmalah together. Bismillahirrahmanirrahim. Step on the following agendas is reciting the Holy Quran, who will be recited by our student, Habib Basanur Murdi, Bachelor of Pharmacy. Habib Basanur Murdi, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. A'udzu billahi minasy syaithanir rajim. Bismillahirrahmanirrahim. ألا إن لله ما في السماوات والأرض ألا إن وعد الله حق ولكن أكثرهم لا يعلمون هو يحيي ويميت وإليه ترجعون يا أيها الناس قد جاءتكم موعظة من ربكم وشفاء لما في الصدور وهدى ورحمة للمؤمنين قل بفضل الله وبرحمته فبذلك فليفرحوا هو خير مما يجمعون قل أرأيتم ما أنزل الله لكم من رزق فجعلتم منه حراما وحلالا قل الله أذن لكم أم على الله تفترون وما ظن الذين يفترون على الله الكذب يوم القيامة إن الله لذو فضل على الناس ولكن أكثرهم لا يشكرون صدق الله العظيم. Assalamualaikum warahmatullahi wabarakatuh.
our student Habib Basha Nur Murdi, who has recited the Holy Quran for us. May Allah always give us guidance and help. Amin, amin, ya Rabbal Alamin. The third agenda is singing national anthem of Indonesia Raya and Mars Muhammadiyah. Please, for the committee to make it ready. And for the participants from our Zoom meeting, please stand up in your position. For the participants, please take your seats. The next agenda is welcoming speech by the chairman of the Ahmad Dahlan International Conference on Health Sciences 2025. Now, I would like to invite Professor Dr. Apoteker Nur Hasanah, Magister of Science. Professor Nur Hasanah, time is yours.
Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. The Honorable Dean of Faculty of Pharmacy, Dr. Apotheker Iis Wahyuningsih, the Honorable Dean of Faculty of Public Health, Dr. Roshidah, and the Honorable Dean of Faculty of Medicine, Professor Dr. Rusdi Lamsudin, our partner from Siam University, Thailand, Ankara University, Turkey, GC Medical University, Japan, Nanjing University, China, and University of University of Kuala Lumpur, Malaysia, and also Japanese Medical University Assembly, JMICA, Japan. Distinguished guests, respected speakers, participants, and colleagues. It is with great honor and sincere gratitude that I welcome all of you to the Ahmad Dahlan International Conference on Health Sciences. On behalf of the organizing committee, I extend our warmest welcome to all participants who have come together today to engage in meaningful discussion, share valuable research, and foster collaboration in the field of health science. This year, our conference proudly carries the theme, One Health, an integrated approach to sustainably balance and optimize the health of people, animals, and ecosystem. This theme reflects a growing awareness and urgent need for a holistic, interdisciplinary approach to global health challenges. The interconnected of human health, animal health, and the environment cannot be overstated. Diseases know no boundaries, whether geographical, biological, or social. And therefore, our response must be equally integrated, collaborative, and sustainable. As we gather here today, we are reminded of our shared responsibility to protect and not only our own health, but also the health of the planet and all its living beings. It is our hope that through this conference, we can inspire new ideas, encourage evidence-based practices, and create a network of professionals and scholars committed to the One Health mission. I would like to take this opportunity to express my deepest appreciation to all speakers and presenters who have kindly shared their time and expertise with us. I also thank our university leadership, academic partners, and every members of the organizing team whose hard work and dedication have made this event possible. May this conference be not just a platform for academic exchange, but also a stepping stone toward real-world solution, impactful research, and lasting partnership. Thank you once again for your presence and participation in this conference. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you very much for Professor Dr. Apotheker Nur Hasanah, Magister of Science, for the greetings. And the next agenda is speech by the Dean of Faculty of Pharmacy, Universitas Ahmad Dahlan. And please welcome Dr. Apotheker Iis Wahyuningsih, Magister of Science. For Dr. Apotheker Iis, time is yours.
Thank you. My voice is okay?
Yes, doctor.
Yeah. Bismillahirrahmanirrahim. Dear speaker and all participants, Assalamualaikum warahmatullahi wabarakatuh. Good morning, dear Dean of the Faculty of Public Health and Dean of Faculty of Medicine and our Varsity Plan. First of all, on behalf of Universitas Ahmad Dahlan, I would like to warmly welcome all participants and distinguished speakers to the Ahmad Dahlan International Conference on Health Science 2025, which is held virtually by the Faculty of Health Sciences at UAD. This year's conference brings to them One Health, an integrated approach to sustainably balance and optimize the health of people, animals, and ecosystem. It reflects our shared commitment for fostering interdisciplinary collaboration in the fields of pharmacy, public health, medicine, and biomedical sciences in addressing global health challenges. I would also like to express our deepest appreciation and congratulations to the entire committee for their dedication and hard work in organizing this event smoothly. Our sincere gratitude as goes to the guest speaker, both from overseas and from UAD, who have kindly agreed to share their insight and expertise with us. Therefore, I would like to personally welcome each of our distinguished speaker, such as Prof. Takashi Yashiro, MD, PhD, from GC Medical University, Japan, as well as Prof. Elgar X from Ankara University, Turkey, Dr. Usasiri Srisakul from Siam University, Thailand, Dr. Fadiasi Dwi Astuti, SKM MSc from Universitas Ahmad Dahlan, Indonesia, Dr. Nur Asma Amin from University Kuala Lumpur, Malaysia, Dr. Yanantri Pingarsip, Ahmad, SG, SPE from Universitas Ahmad Dahlan, Indonesia, Uyangi Putsaka, MSc in Farm, BCPP from Siam University, Thailand, Asif Prof. Onjun Mehmet Saka from Ankara University, Turkey, Wang Zizi, PhD from the second affiliated Hospital of Nanjing Medical University, China, and Dr. Apotheker Adnan MSc from Universitas Ahmad Dahlan, Indonesia. Ladies and gentlemen, Alhamdulillah, this event has brought together hundreds of enthusiastic participants from various countries and backgrounds, both nationally and internationally. Thank you for joining us in this academic gathering that will be held virtually via Zoom on May 20, 2025. We also extend our sincere thanks to Universitas Ahmad Dahlan for their continuous support in facilitating this international academic event. Although this offering is conducted online, it serves as a bridge for knowledge exchange and collaboration across countries and disciplines. Certainly, this conference offers a wealth of benefit from expanding international network, encouraging future research partnership to strengthening global academic communities. Finally, we truly hope that you will enjoy this conference and that future programs can be held in person here in Indonesia with even more comprehensive preparation. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Okay, thank you very much for Dr. Apoteker Iis Wahyuningsih, Magister of Science, for the speech. But ladies and gentlemen, before we go to the next agenda, please let us to take a picture in our Zoom meetings. I would like to call slides by slides, and please for the committee to help me to screenshot the Zoom itself. Okay, we start from the slide 1. Okay. Okay, thank you. Then, okay, then the slide 2. Okay, please open the camera and then give the big smile for us. Okay, thank you very much. Then the third slide. Okay, that's amazing. There are a lot of people there. Okay. Then slide 4. Okay. Then slide 5. Okay, and the last for the last slide. Yeah, okay. Thank you very much for the participants. And then let's skip to the next agenda. This is the presentation from our great keynote speakers. But we've got an exciting lineup of the speakers and the event plan for today's conference. Actually, we hope you will find it engaging and also informative. So let's get started. Actually, today's conference serves a dynamic platform for researchers, academics, professionals, and policymakers to exchange knowledge and insight on the current hot issues through one hot perspective. Actually, this integrated approach emphasizes the interconnectedness of the human, animals, and also the environmental health. The first Ahmad Dahlan International Conference on Health Sciences, with the theme of One Health, an integrated approach to sustainably balance and optimize the health of people, animals, and ecosystem, organized by the Faculty of Pharmacy, Faculty of Public Health, and Faculty of Medicine of Universitas Ahmad Dahlan. And it is co-hosted by the Siam University of Thailand, Ankara University of Turkey, and Jichi Medical University of Japan, Nanjing Medical University of China, and University of Kuala Lumpur, Malaysia, and also GMICA, Japanese Medical University International Collaborative Assembly. We have some distinguished speakers lined up who will be sharing their insight and knowledge on this topic today. But so, I urge you to sit back and then relax and absorb the information that will be shared for today. It is promises for us to be an insightful and also enriching experience for all of us. And I hope that by the end of this conference, we will all have a better understanding to collectively share information that can be used to address the recent health issues and challenges. Before we start our conference, allow me to invite Dr. Leonie Dwi Rizkita, Magister of Biomed, as the moderator for this day. For Ms. Leonie, are you with us already? Okay, thank you very much, Ms. Leonie. And before that, I would like to read the Ms. Leonie short profile. Ms. Leonie actually graduated from Magister Degree of Universitas Ahmad Dahlan, Indonesia. From Universitas Gadjah Mada, Indonesia, and she is currently working as head of pharmacology department of the Faculty of Medicine, Universitas Ahmad Dahlan, and she is a lecturer of Faculty of Medicine of Universitas Ahmad Dahlan itself. Her topic interest is all about the medical pharmacology, and she also has a lot of representative research and related to this event today. So, are you ready, Ms. Leonie? Okay, time is yours.
Good morning to all distinguished participants who have joined us in this Zoom room today from all across the nations. It's my great honor and pleasure to welcome you to this session of Ahmad Dahlan International Conference on Health Science today. Allow me to introduce myself. My name is Leonie Dwi Rizkita. You might call me Leonie, and I will be guiding you to this first keynote speaker today. This session aims to foster cross-disciplinary dialogue and to present evidence-based strategies that can be applied both globally and locally, especially research on medical histology and biology. Our first speaker is Professor Takashi Yashiro, MD, PhD, a professor emeritus from Jichi Medical University School of Medicine, Japan, who has been well known for his research on medical biology and histology, especially about anterior pituitary cells. His presentation for today's keynote speaker is titled Current Research Methods of Anterior Pituitary Cells in the Field of Medical Biology and Histology. Professor Takashi Yashiro, MD, PhD, has published extensively on pituitary experiments with various approaches, especially in cell morphology and characterization. And before we move to our keynote speaker session, I would like to read and show you the curriculum vitae of our first keynote speaker. Professor Takashi Yashiro, MD, PhD, has graduated from JK Medical University School of Medicine at 1978, and he obtained his PhD at 1984. Currently, he became the Director of Nursing School of NASU, and at 2010, he got the Yoshimura Prize of Japan Society for Pituitary Research. What a wonderful pleasure to allow me to join us with us, Professor Takashi Yashiro, MD, PhD.
Okay, thank you very much. Can you hear me?
Thank you.
Okay. Well, first of all, I would like to say thank you very much for your wonderful introduction. And can I share my slide by myself?
Yes, you can explain, and yeah, the presentation has already been aired on, yeah.
Now you see my slide? No problem.
Okay.
Okay, good. And also my voice itself, it sounds good. No problem.
No problem.
Okay, thank you very much. I would like to start my talk today. And after my talk, I would like to have the QA session. Okay, at that time, if possible, I would like get my question from other participants, if possible, using the chat. Understand? Because I had many chances in the past to talk on Zoom and have a discussion, but sometimes I had this difficulty to hear the voice itself. Sometimes bad voice or confusing. So the chat is best for me. Okay, thank you very much. Anyhow, thank you for your introducing me. I would like to appreciate it. And first of all, I say congratulations for your holding wonderful and meaningful international conference. First, Ahmad Dahlan International Conference on Health Science. Congratulations. And it is also my great honor and pleasure to have a wonderful time to present my talk today here on Zoom. Before starting my talk, I also would like to express my thanks to the conference committee members, especially Dr. Professor Nora Hasana, chairperson of this international conference, and the committee members, especially the moderator, Dr. Leonie. Thank you very much. Well, my today's topic talk is current research methods of anterior pituitary cells in the field of medical biology and histology. And the research method itself was done in the laboratory anatomy of my Medical University. This is my CV. After graduation from medical school when I was young, I directly started my research and education career. GK Medical University, my graduated medical school, which is an oldest medical school in Japan, established about 150 years ago, so old one. I entered the Department of Anatomy, Department of Histology, and I have been working for GK Medical University more than 30 years. In 1998, it is my life, luckily got the position of the professor and chairman of the Department of Anatomy, Histology at GK Medical University. And after retirement from the position in 2018, I started my work at other medical school or other nursing schools. And of course, I have still communication with my old laboratory department. Department of Anatomy, Division of Histology and Cell Biology of Japanese Medical School. We have in Japan 82 medical schools. Our GK Medical University School of Medicine is just one of them. Introduction of our laboratory. Please look at the photo. This is the laboratory members about 8 years ago. Many researchers coming from Indonesia, Rita, Topi, and Dini here, Imi, Dini, Topi, Rita. They are from Andras University and the University of Indonesia, and she's from Mongolia. The other members, of course, Japanese, including me, all members, had been researching pituitary cell function. This is a textbook drawing of the anatomy of pituitary gland. As you see it, here is anterior pituitary gland, the posterior gland. Usually we call it pars discalis. In narrow meaning, this is anterior pituitary gland. And first, what kind of cells exist in the gland? As you see, GA cells, somatotroph, TSA cells, cytotroph, ACTH cells, corticotroph, prolactin cells, lactotroph, ADH cells, gonadotrophs. And these cells secrete their own hormones. In addition to these, this will be the key cell, follicular stem cell. So this is, so to say, supporting cells of the anterior gland. Also S100 protein is marker of follicular stem cells. The pure function of follicular stem cells still unknown. Anyhow, actually act as supporting cells. Well, and these cells' function is, of course, mainly, strongly controlled by the hypothalamic hormones. Understand? For example, the TRH in the hypothalamus released into the portal vessel and go into the anterior pituitary port and act on the thyrotroph, stimulate thyrotroph, and secrete TSH, thyrotropin. And thyrotropin, TSH, go into the thyroid gland and stimulate the release T4 and T3. And when the serum level of T3, T4 goes up, it's negatively feedback to the thalamus. This is, everyone knows, hypothalamus pituitary periphery axis. In addition, in our laboratory, we have been studying more pituitary function regulation system. The project topic is like this. For example, cellular function regulation by ECM, extracellular matrix, roles of follicular cells in functional tissue, roles of cell adhesion molecules, roles of the locally synthesized retinoic acid in the gland. Anyhow, in one word, pituitary cell function with regulation system. And to perform this kind of researching project in our laboratory, we have these analyzing ways or research instruments or equipment: confocal laser microscopy, fluorescence microscopy, electron microscopy, immunohistochemistry, RT-PCR, real-time PCR, Western blotting, and FACS, fluorescence activated cell sorting. It's very important, sorting equipment. And LMD stands for laser capture microdissection. We use these kind of ways to make the analyzation of pituitary cell function. Confocal laser microscopy observes the cells in the thick section, or we can observe the cells, culture cells, as it is. We don't need to make any thin sections. We don't need to make slices. And we have the standard type microscope and converted type scope. Also, 2-photon. It's high ability 2-photon system microscopy. As you see, nowadays it's widely spread to use. For example, this is Olympus one. This is the standard type, and this is converted type to observe the culture of cells. The principles of the confocal laser microscopy started in 1960s, and 2 years later, from 1880s, the confocal microscope itself is start to use. Next, electron microscope. We can observe the fine detail of ultra-thin specimens when we use electron microbeam. And as you see, there are 2 types. This is transmission type. For example, this is right under pituitary gland. We can observe more than 20 cells in low magnification. This is the capillary lumen, the nucleus of the hormone-secreting cells. In a little higher magnification, this is capillary lumen, this is pericyte, and we call it gonadotroph, which produce LH and FSH. Please look at small black granules. These are secretory granules contains LH or FSH so much. And besides granulocytes, often we encountered the production cells. Production cells embrace LH cells. It's a very interesting phenomenon for morphologists. In the case of the GS cell, this is typical GS cell, somatophores, secret the somatotropin. This is characteristics of the SHTH cells by structure. Look at secretory granules arranged along the cell membrane. This is the characteristics. And ACTH cells and GS cells, very good friends each other, always live together. This phenomenon is also very interesting for us. Well, nowadays, we can apply this electron microscopic images to make the 3D reconstruction of cell organelle. For example, like this. Look at axon and mitochondria in the axon. Nerve axon and mitochondria's localization in the axon. These images also very interesting. They give us many ideas to make research of the function and morphology of the axon or nerve cells. Okay, I'd like to show the scanning electron microscope. We can observe the fine topography of the specimen, of course using electron beam. This is the anterior pituitary gland. When we observe using the scanning electron microscope, I would like to use this slide again later. Anyhow, this is the capillary, and this is the pericyte. Notice appearance of the immunohistochemistry greatly contributed to the development of biomedical science. Of course, we have two ways. One is dark method, one is light method. In addition to immunofluorescence method, we have light field way immunohistochemistry, mainly called labeled antibody method. This method was developed by the Professor Nakane. This is the principles of the immunohistochemistry, labeled antibody method. He is Professor Nakane. I personally respect him very much. And it was about 10 years ago when I held the Japanese anatomist meeting all over Japan. I called him as a keynote speaker of this conference. I would like also introduce one more very important person. As you see, we often use this kind of coloration, fluorescence, green and red. He is Dr. Osamu Shimomura. He is the first person to found and discovered the green fluorescence protein. He got the Nobel Prize for his achievement. I had a good chance to hear his lecture, and after that, I likely had a chance to talk with him. He's very smart, very kind, very honest, wonderful character. I also respect him very much. Well, I would like to show 2 or 3 immunohistochemical slides. It was then when I was very young. Now I am 70 years old, but when I was 30 to 35 years old, I made this slide. Double immunostaining by use of Nakane's labeled antibody method. In one section, I stained LH cells and prolactin cells. Blue or little violet. This is gonad of LH cells. And the brown one, prolactin cells. Instead, please check the combination of LH and ACTH cells. Also, this is LH cells. And the brown one, this is ACTH. They never exist each other separately, always. But in the case of LH and prolactin, they live together, good friends, like the relation I and my wife. It's very interesting. I also show you this picture was taken about 40 years ago. Oh no, 30 years ago. These sections are serial section. First section and second section, serial. On the left, I stand LH beta. On the right, I stand FSH. Look at this cell positive against LH beta antibody. And this is the same cell. Same cell, yes. The positive against FSH beta. That means this cell produces and secretes 2 kinds of hormones, 2 different hormones. One is LH, one is FSH. After that, gonadotroph. Of course, we can still use this name gonadotrophs, but it's much better to express LH per FSH cells like this. Understand? LH by FSH cells. It's much better. We can apply the immunohistochemistry at the EM level. There are 3 ways. One is pre-embedding method, immunostaining of LH beta. Result was shown like this. And when we use the post-embedding method by use of the very small colloidal gold, the colloidal gold localized mainly on the secretory granules. This is the evidence. The LH localized in the secretory granules in the cytoplasm. Okay. When they use ultra-cryosectioning, as a technique, it's very, very hard. Not everyone can do that. A specially trained person can do that. But cryoimmunostaining shows higher and higher specificity. Look at here in higher magnification. Gold particles clearly localized only in the secretory granule. Very good technique. To detect the protein, as you see, the development of the immunohistochemistry is very helpful for us, the morphologists. But in addition, this is a way to detect specific mRNA in the section. In addition to immunohistochemistry, in-size hybridization method is very useful and widely used. to detect mRNA on the section. The technique itself is similar to the labeled antibody method. In the place of the antibody, we use de-labeled probe. And this is the result. As you see, the brown color shows the localization of mRNA of LH beta in the section. Okay. And to note the section, of course, to detect mRNA, easiest way is use the RT-PCR. We can easily detect mRNA expression. Even the medical student can do this technique. But when you quantify the mRNA expression, we have to use real-time PCR. Anyway, both of them are now commonly used in the world, even in the Department of Anatomy. This is also the very old technique, Western blotting, to detect the measuring the messenger level, and in addition to measuring the messenger level, we can easily check the protein itself. Detection and quantification of certain protein is now very, very easy. Do you know the facts? It's a very useful technique to separate specific cells. Fluorescence activated cell sorting. For example, the cell sorting, cell counting, biomarker detection by laser. This is for cell sorting by use of biomarker detected by laser. For example, GFP positive cells, you can easily collect only the positive cells, and negative cells goes this one. This is the histogram. We can collect only the positive cells or only negative cells. This is a very useful way. For example, to collect only follicular cells from the anterior pituitary cells is not difficult when we use this sorting system. In the LMD, laser capture micro dissection, it is much more interesting. It's similar to the usual type of microscope, but, for example, we see the culture cell like this. We find the region, for example, we focus on the cell type and select the region, this spot. And by use of the laser, we can cut here, dissect here, and move to other place. So we can select, isolate small area or this one, just one cell, and collect them for analysis. There are many analysis ways: inside hybridation, Western blotting, PCR, anyhow. So, like this, using these updated analytic ways or tools, we are starting a pituitary gland function in the laboratory. So then, we'd like to show you just some result, some data of our laboratory. This is kinds of textbook drawing importance of the local communication in the gland. As I explained at the first slide or second slide, of course, pituitary cells is directly controlled by hypothalamic hormones in the system of hypothalamus, pituitary, and periphery. But our laboratory has been studying for a long time about local cell-to-cell and cell-to-extra matrix communications and their molecular mechanism, including paracrine, autocrine, matricline, juxtacrine regulations. Matriclines is an extracellular matrix action, and juxtacrine is a direct cell-to-cell interaction mediated by membrane proteins. And this is also the general idea of the ECM effect on the cell function. In addition to soluble signals, for example, growth factors, cytokines, and so on, in the case of pituitary gland, hypothalamic hormone is one of them, and a strong one. And physiological signals, ECM, like this one. Physiological signal of ECMs act on the cell and influence cell behavior in each organ and tissue of the body. And what happened in the pituitary? What is happening in the pituitary? Cell-to-cell function, ECM function, local communication. These are our researching project. I would like to show you some data. This is live cell imaging of type 3 collagen-coated surface. Okay. Usually, when we perform the culture study, we use non-coated plastic dish. When we coat, in this case, type 3 collagen, and start to culture, what happened? Again, by use of time-lapse imaging, we succeeded in showing a pituitary proliferation for the first time. When we culture the cells on the ECM-coated surface, in this case, type 3 collagen, follicular starter cells often show their proliferation by cell division. Please understand, this is the first live imaging of anterior pituitary cell proliferation like this. Live imaging. Yes. And next, what happened to the other ECMs? Collagen 1, 3, 4, laminin, proliferation rate is checked by BLDU uptake experiment. And as you see, we could see about 100% increasement when we culture follicular cells on every kind of ECMs, like this. Okay, I would like to move to the topic of the gap junction. As you know, the gap junction exists between the cells. It's the communication way of A and B cell is very important. For example, the muscle cells, they should have the gap junction between the cells. And the gap junction is very important for the simultaneous action of the contraction of the muscle cells like that. In the pituitary gland, especially between the follicular cells, there is a connection. Gap junction. We check the difference of gap junction formation between cultured or non-cultured coated, human-coated surface lamin. As you know, the connexin 43 is a major protein composing gap junction. Then we examine the fine structural changes of follicular cell cells. The first, this is control. We culture the cells on non-coated dish. Gap junction can be seen at red signal. And when cultured cells on laminin-coated dishes, as you see it easily found, the red signal increased. At mRNA level, yes, is about more than 100% increasement. What happened to the plasma level also increased about 60%. So by use of the immunohistochemistry, Western blotting, and the RT-PCR, we examined very precisely about the changes of gap junction formation, and also observed electron microscopy. This control non-coated one. This is laminin-coated one. This circle is dispersed, enlargement dispersed. Please look at here, the controlled Golgi apparatus. But when we culture cells on laminin-coated surface dish, Golgi apparatus shows very, compared to two of them, highly developed. They also, yes, red arrowhead shows. This is the gap junction. Understand? Very interesting. When we cultured on the laminin-coated surface, cell organelle including Golgi apparatus, as well as gap junction itself, present well development. Actually, this shows the electron microscopical data also showed the ECM act on the cell, stimulate the cell. Okay. Next, analysis effect on laminin on LH release. I'd like to show you a typical example of ECM action on pituitary cells. I'm sorry, this is unpublished data. The effect of laminin on LH release from glucose. We use a primary monolayer culture and disperse cells, and after 40 hours incubation, we add the laminin into the medium. Okay, and what happens? Yes, LH release into the medium increase dose dependently 3 to 4 times higher than that of core. So here there is a big question. What cell type in pituitary gland produce many kinds of ECMs? Laminin, collagen, what type? So many people say fibroblast. But unfortunately, it is well known among morphologists, fibroblast never exists in the pituitary gland. Then we move to find out which cell type produce ECMs. I previously showed this slide. This is a capillary pericyte and the part of the FS cells. As to the detail, I would like to cut it, but the result is yes. In size hybridization reveals type 1 and type 3 collagen produced by pericyte. Is this side. This side produces collagens in the pituitary gland. Furthermore, Through the examination detecting collagen-producing cells, we could find new cell type that have never reported in the past. This is novel desmin immunopositive cells, deep cell. This cell type. This is not fibroblasts. The deep cell locates in the perivascular space without basement membrane and showing a little large and polygonal cell shape. This cell type also produces many kinds of ECMs. For example, fibromodulin, lumican, leucorin, osteoglycan, like that. Through these kind of study, for example, in this paper, we found this kind of collaboration system exists in the gland. For example, pericyte produce type 1, type 3 collagen act on the follicular star cell. And this cell type release TGF-beta 2 and stimulate the pericyte itself. This kind of communication system, local communication. Also, I would like to cut the detail of that. The local communication of FS cell, ADS cell, laminin, matricline. These are published, these papers, even in human pituitary. Almost the same phenomenon is occurring. So this may be the almost the final slide I'd like to use again. There is, of course, hypothalamus pituitary periphery acts as strong control system in the gland. In addition, in the pituitary gland, inside of it, we have paracrine autocrine system, matricline system, extracellular matrix act on the cell that causes phenotype changes. And the juxtacrine, cell-to-cell intact, mediated by the cadherin. And these stimulate and change the cell phenotype from production, cell network, regulator, progenitor, niche, like that. So again, I'd like to use this slide. Using these updated analyzing with tools, we are studying pituitary gland function in the laboratory. The conclusion of today, even in anatomy and histology laboratories, it should be normal for research to be conducted not merely by the morphological method, but by the multifaceted method, physiological, biochemical, and molecular cellular level. By doing so, new findings will be easier to acquire. Thank you very much for your attention. Time is okay. Thank you.
Yes, thank you very much for your presentation, Professor Takashi Yashiro, for that insightful and timely presentation. Your presentation is very wonderful, and it shows us about the cells imaging.
I'm sorry, I cannot hear your voice.
The voice. Thank you very much for your presentation, Professor Takashi Yashiro, for that insightful and timely presentation. It's a very wonderful presentation of cells imaging you showed to us in regards the methods and tools to explore the visuality and quantification of cells in pituitary cells. Now we are going to move to our Q&A session. We have 15 minutes, and please, for all the audiences who want to ask to Professor Takashi Yashiro, you can type on our Zoom chat box, or you can raise your hand, but I will suggest you to text on the chat box. Okay, while we wait other audiences who wants to give questions to you, can I give you some questions, Professor?
Yes.
Yes, I am so thrilled and enticed by your presentation in the early slides that you have a very few current research topic in your laboratory. Since my major is in pharmacology, I am so excited about then how you choose about the synthesized retinoic acids and the effects of the anterior pituitary cells. I wonder, is there any reason in choosing the retinoic acid and observe the effect on the pituitary cells?
Would you please, at last part, would you please again?
Yeah, I'm asking about in the very first slides of your presentation, you mentioned about the synthesized retinoic acids in the anterior pituitary cells. I wonder, is there any reason in choosing the retinoic acid and observe the effect on the pituitary cells?
Oh, yeah, okay, understand. Just a moment, please. This one?
Yes,
Yes.
it's visible?
Today, I do not have enough time to explain about this study. But using microarray system, we found that retinoic acid it is synthesized in pituitary gland. So what kind of action do they have? Yes, we started very well. They have some specific action. So when you have a time, please follow our papers published in the past. We have published about more than 5 papers as to the function of retinoic acid. Okay.
Okay, thank you very much for your answers. Now we have some questions in the chat box, Professor. If you may read also in your chat box, there is Miss Sarifatul Mufida, who asked about the confocal images of growth hormone localizations. And she asked whether, is there any major difference in the sample preparation, staining protocols, or imaging settings that you have found critical for getting such clear images in growth hormone studies?
Yes. As to the preparation of slide, and most important thing is when you use confocal microscope, and you are going to observe the cells in the culture or the section. In the case of the culture cells, when you try to observe the culture cells, somatotroph or other cells, the most important thing is you should obtain the good culture good quality or high quality of culture itself. Understand? When you use when you use the primary cells or the cell line cells, either way. And when you observe the sick cell, of course, you had obtained the good quality, high quality of the fixation or the embedding method. Cutting technique is not so important when you use a confocal micro laser scope. Understand? So maybe most of the researchers use the confocal microscope for observing the culture cells. So the culture technique is very, very important.
Okay, thank you, Professor.
Okay. Yeah.
Is it clear, Ms. Sharifatul?
Yes, thank you.
Yeah, thank you very much. Professor, we also have another questions in the chat box. It is from Miss Anissa, Mrs. Anissa, and she asks whether about the staining methods can be done even with limited facility in the institution. She wondered if we can compare the cryoimmunostaining with the conventional immunostaining for research objective and methods. And she wondered if there is any specific immunostaining that we could use as simple as possible with limited resource and facility.
A very important question. Thank you very much. It's
Yes,
a nice question. Yeah, very nice question. She would like to know the difference of the technique and the result of cryoimmunostaining and the conventional immunostaining. Everyone knows when you use the conventional ones, the quality of this image is higher than that of the cryoimmunostaining. And when we look for the higher immunospecificity or immunoreactivity, cryoimmunostaining is much better, show the good result compared to the conventional one. So I would like to make two advices. One, if you need the immunohistochemical specificity, or if you need the immunohistochemical reactivity itself, you had better use the cryoimmunostaining. When you prefer to obtain the good images, you had better use the conventional ones. Okay, this is my first advice. And second advice, to make good quality cryosection, if you can make it, it's better. Understand? Thin section, for example, the 4 micrometer cryosection. You can make it, and please use the new fresh knife. It's very, very important. Okay, I can give you the two advices. This is a very good question. Thank you very much.
Thank you very much for Takashi Yashiro for the answer of your questions. For the question from Mr. Anissa, so I can summarize that it's very important to choose our tools, and instead of using the old ones, we have to choose the new fresh knives to obtain a better result of the tissue dissections. And since we are limited by time, and I have to make conclusions of our first plenary sessions in today, Professor Takashi Yashiro has extensively conducted research about the pituitary experiments with the various approach in the cell morphology and characterization. And he also showed us various methods and tools that we can apply for many kinds of tissue dissections and tissue morphology observations. And before we close our keynote speakers on today, I would like to share and give you, Professor, the certificate through the online Zoom. Please accept our certificate for becoming our keynote speaker for today.
Thank you very much. Please send it to me using email.
Yes, absolutely. We will going to send it through your email.
Okay, thank you very much. Anyhow, I would like my great thanks to moderate Dr. Leonie.
Yes.
Thanks a lot. You're a good controller. Thank you very much.
Thank you very much also. Very welcome for me too. And since we have closed our first keynote speaker, I would like to return my sessions to the MC and going to continue with the other keynote speakers. Thank you very much.
Thank you.
Okay, thank you very much, Dr. Leonie, for being the moderation, and also thank you very much for Professor Takashi Yashiro, a philosophy of doctor from GC Medical University. Actually, this is impressive presentation from our keynote speakers on the first time, and then we are really thank you for being part of this conference for this day. And then before we go to the next session, let's give another round of applause to Professor Takashi Yashiro, Philosophy of Doctor, for sharing such valuable knowledge and also the things that's so important for us. Please give big applause for Professor Takahashi Yashiro. Okay, thank you very much for the participants. Okay, then we go to the next session. The next agenda is the presentation from second keynote speaker, Associate Professor İlker Ateş from Ankara University of Turkey. But before I start our conversations, allow me to invite Apoteker Lalu Muhammad Irham, Magister Farmasi, Philosophy of Doctor, as moderator for this day. Mr. Lalu, are you with us already?
Yeah, Miss Putri.
Okay, thank you very
Yeah,
much.
ready.
But before that, I would like to read Mr. Lalu's short profile. Actually, Mr. Lalu graduated doctoral degree from Taipei Medical University of Taiwan, and he is an alumni of Universitas Ahmad Dahlan too, especially in Faculty of Pharmacy. He is currently working as head of scientific publication, and he is a lecturer of Faculty of Pharmacy, Universitas Ahmad Dahlan. His topic interest is all about the pharmacogenomic, bioinformatics, precision medicine, cancer genomic, and also drug repurposing. He has also a lot of representative research that related to this event today. So are you ready, Mr. Lalu, to be the moderator for this day?
Yes.
Okay, Mr. Lalu, time is yours.
Thank you so much, Bu Putri, for kind introduction. Assalamualaikum warahmatullahi wabarakatuh. Welcome to our event, Ahmad Dahlan International Conference on Health Science 2025. Good morning, everyone. Good morning, Professor İlker Ateş. Are you on Zoom?
Good morning, Dr. Irham.
Yeah, thank you so much, Professor İlker Ateş. How are you, professors?
Thank you very much. I hope you are all fine.
Yes, thank you so much. First of all, I would like to thank Allah for giving us health and opportunity to attend our event this morning. Secondly, salawat and salam to our Prophet Muhammad sallallahu alaihi wasallam for his guiding from the darkness to the lightness. The Honorables head of today's events, Professor Nur Hasana. The Honorables, as well our speakers in the first sessions, or in the second sessions, Professor İlker Ateş from Ankara University, Turkey. Last but not least, I would like also to thanks to the participants for attending our webinar for today's event. Before we start, our speakers for this event is going to be our Professor İlker Ateş from Ankara University. He will share to every single of us here about the use of biomarker in deciphering human health impacts on environmental contaminants. Let me share screenshot profile of Professor İlker Ateş. Professor İlker Ateş is professor at Ankara University, Turkey, as Gazilik Faculty. He was born at Ankara in 1976. After completing his high school education at that Ankara College in 1993, he started his university education at Ankara University, Faculty of Pharmacy in the same year. After completing his undergraduate education in 1997, he started his master's degree in Ankara University, Faculty of Pharmacy, Department of Pharmaceutical Toxicology in 1998. After completing his master's degree with thesis in 2000, he received his doctorate in the same departments between 2000 and 2005. In September 2013, he was awarded the title of Associate Professor in Pharmaceutical Toxicology. He continues his work as faculty member at Ankara University, Faculty of Pharmacy, Department of Pharmaceutical Toxicology. He has published nearly 60 international and national respected peer-reviewed journals with 250 citations, many projects in which his work as researcher and executive, and has been a book chapter, author, and translator. In addition, there are many national and international congress and symposium proceedings related to his field of study. He has also been serving as the Peace Dean of Ankara University, Faculty of Pharmacy since September 2019. Professor İlker Ateş, are you ready to deliver your speech?
Thank you for your great introduction, Dr.
Yeah.
Irham.
Thank you, Professor Ricardo. You will have around 45 minutes to deliver speech, and we have around 15 minutes for Q&A sessions.
Okay, okay. Let me share my screen. I hope it is seen, my slides.
Yes.
Okay, let me start. Thank you for giving this great opportunity for me to talk about my lecture in this great conference. So let's start. I will talk about the use of biomarkers in deciphering human health impacts of environmental contaminants. Now, let's talk about a little bit with the biomarkers. A biomarker is a measurable substance, typically a chemical or biomolecule, such as a protein, lipid, nucleic acid, found in biological samples that's the indicative of a normal or abnormal condition of a living organism. So that this may be the question, how can biomarkers be used in deciphering human health impacts of the environmental contaminants? So biomarkers have long been used in medicine to diagnose and monitor disease progression and response to therapy. They are also a critical tool for evaluating human exposure and response to chemicals in the environment. Diverse biological samples are used by environmental health scientists to measure biomarkers in humans, including blood, urine, and other bodily fluids, tissues such as adipose, bone, teeth, and hair, and the images of living tissues. As you see from the table, there are examples of matrices commonly used to assess biomarkers of exposure and effect. The biological matrices can be called as breath, saliva, or sputum, or urine, or feces, or hair, nails, and sweat, and it goes on, such as nasal swab, breast milk, blood, teeth, umbilical cord blood, placenta, amniotic fluid, adipose tissue, and also bones. So what are examples of exposures detected? So maybe carbon monoxide from the breath, or nicotine or thiocyanate from the saliva or sputum. Or, as you know, the urine is the main matrix of the example of exposure, such as the toxic metals or ochratoxin or cotinine. So that
Apa saya iniin? Ingetin.
Okay, so that there are lots of examples of exposures detected. And what are the examples of health effects or outcomes monitored? When we look at the breath, some diabetes or ketosis or lung cancer or asthma can be seen. Or from sputum, oral cancers or periodontal disease can be seen. So that we can expand these examples from these slides. So I don't want to read all of them one by, but as you see, there are lots of matrices, there are lots of examples of exposures detected, and lots of examples of health effects. As we go on, maybe from the blood, we can see some various toxic metals, and these can cause hormone balance problems or cholinergic crisis. So what has the ideal biomarkers has some specifications we want from the ideal biomarker. It must be a quantifiable, or it must correlate to clinical outcomes, or it must be sensitive and specific, or it must be cost effective, and it must be personalized management. So that there are lots of features we want from the ideal biomarkers. And when we look at the classification of biomarkers, as you see, there are lots of subgroups, such as gene-based or protein-based or disease-based or metabolism-based or source-based, so that the classification can be expanded like this with some subgroups.
Pardon, Professor. Could you
Biomarkers
your
used in the environmental health studies are broadly classified as biomarkers of exposure, biomarkers of effect, and
Pardon,
biomarkers
Professor?
of stability.
Professor
Sorry?
Iker, could you show your slide, because the slide is not going to
Okay,
The next slide. Yeah.
let's. Is there a problem? So
Udah.
let me share my slides also. Now, is it seen?
Yes. Could you
But
slideshow?
it isn't going on, so that. What is the problem? So let me open again. Is it going on?
Yes. Yes.
Okay.
Let's work now.
Do you want me to repeat again or go on? I was here. The classification of biomarkers.
Screens to slideshow your slide, Prof.
Now, is it seen?
Yes,
The
but
next one. Okay, the biomarkers
yes.
classification. Okay, let's go on like this. I think everything's okay now. So the biomarkers used in the environmental health studies are broadly classified as biomarkers of exposure, biomarkers of effect, and biomarkers of susceptibility, as you see from the slide. So there are different types of biomarkers used in environmental health studies. Now let's explain one by one. The first one is biomarkers of exposure. So that these are used to evaluate whether an individual has been exposed to a chemical, and if so, how much. And these are used in occupational settings to ensure that workers are not exposed to unsafe levels of chemicals in the workplace. And they are also used in epidemiological studies to evaluate associations between exposure to environmental chemicals and adverse human health effects. The use of these enables epidemiologists to rank subjects according to their exposure level and determine whether the health outcome is significantly correlated with the magnitude of exposure. And when we look at the biomarkers of effect, they are used to measure biological responses to chemical exposures. So they are used in the workplace to monitor the health of workers with potential occupational exposures to toxic chemicals, and in epidemiological studies to assess health conditions in exposed versus unexposed populations. For example, altered blood levels of a liver enzyme as a biomarker of liver toxicity. So biomarkers of effect that are based on the molecular mechanisms by which the chemical causes toxicity can also provide insights on how chemical exposure influences adverse health outcomes, such as biomarkers of mitochondrial dysfunction has been used to identify chemicals that increase the risk of Parkinson's disease by interfering with the function of mitochondria. In some cases, a biomarker can be a measure of both exposure and effect. The example is the activity of acetylcholinesterase in the blood. This is a good example, so that it is an enzyme that inactivates the neurotransmitter acetylcholine, as we all know, to terminate signaling between upstream and downstream cells in a neural circuit. So organophosphate insecticides cause neurotoxicity by inhibiting this enzyme in the nervous system. Thus, acetylcholinesterase activity in blood samples is widely used to monitor exposure to organophosphate pesticides in agricultural workers and to confirm the organopesticides exposure in individuals exposed to these compounds either accidentally or intentionally. Because this biomarker measures a biological response to OPS, it can also be considered as a biomarker effect, and the magnitude of the enzyme inhibition in the blood is used to predict the likelihood that an exposed individual will develop severe symptoms of organophosphate poisoning. So the next biomarkers of susceptibility are used to assess an individual's vulnerability to the toxic effects of chemical exposure. These can include genetic factors that increase or decrease the toxicity of environmental exposure or other biological factors, such as metabolism, diet, age, and health status, that influence individual response to chemical exposure. For example, elevated cholesterol levels, which are associated with an increased risk of developing numerous diseases following toxic exposures. So these are also useful for understanding gene and environmental interactions involved in disease pathogenesis and provide potential answers for explaining the variable responses often observed among individuals in an exposed population. So this is a good slide to summarize these biomarkers of effect, biomarkers of susceptibility, and other biomarkers of exposure. So from now on, I will discuss examples of well-established and emerging biomarkers used in environmental health to assess exposure to and effects of chemicals that target the gut, respiratory system, and brain. The gut and respiratory systems are the first organ systems impacted following exposure to environmental chemicals via ingestion or inhalation, which are the primary routes of human exposure to chemicals. A number of non-invasive or minimally invasive approaches are available to assess environmental impacts on these organ systems. In contrast, there have historically been few to no biomarkers for assessing environmental impacts on the living brain. However, recent advances in technology for imaging the living brain suggest exciting new approaches for overcoming that challenge. So the first group is the biomarkers associated with the dietary exposure and toxic effects on the gut. Humans are commonly exposed to toxic contaminants via ingestion of contaminated food and water, as we all know. The gut is a primary line of defense against these contaminants. The mucus-coated epithelial cell lining of the gut provides a physical barrier that restricts uptake of many substances, while diverse enzymes in the gut can metabolize toxic substances to make them less toxic or easier to eliminate from the body. So another important defense against toxic substance in the diet is the community of the microorganisms that live in the gastrointestinal tract, also known as the gut microbiome. The gut microbiome plays a major role in the human health by aiding in digestion, releasing nutrients, and producing metabolites that are taken up into the circulatory system and distributed throughout the body to influence the health and function of organ systems. The gut microbiome also facilitates the breakdown and the elimination of toxic substances in the diet. The waste material produced by the gut and the gut microbiome is eliminated as feces. Thus, stool samples are an important source of biomarkers of exposure to environmental contaminants. So in this slide, we can see that toxic chemicals can readily enter the food chain as exemplified by pesticides. Experimental animal models have shown that the environmental chemicals in the diet can change the types of microbes and associated metabolites present in the gut microbiome. So stool samples can be analyzed for specific environmental contaminants on their metabolites, for example, pesticides, heavy metals, persistent organic pollutants, and more recently, nowadays, micro and nanoplastics. However, this approach will yield exposure data only for specific substances that are being measured, and it is well known that we are exposed to tens of thousands of different environmental chemicals in our diet. An emerging approach for generating a more holistic assessment of dietary exposure to the toxic substances is to measure the diversity of microbes in the feces. Gut microbes are continuously shed in the feces, and both human and animal studies indicate that the profile of microbes detected in the feces also reflects the profile of microbes in the gut itself. Experimental animal models have also demonstrated that dietary exposure to environmental chemicals can change the microbial composition and metabolites in the gut microbiome and feces as mentioned in the last figure. So the limitation of this approach is that it doesn't identify specific dietary substances causing the change. It's hypothesized that the different classes of toxic chemicals generate a unique microbial fingerprint. For example, there are chemical-specific shifts in microbial diversity or metabolite profile, but this has yet to be proven. While the gut typically functions to defend us from harmful environmental contaminants in the diet, it can be adversely impacted by dietary exposures. For example, dietary exposure to arsenic is linked to intestinal inflammation and cancer, while PFAS in the diet is associated with significant gut dysbiosis. So imbalance of the gut microbiome is linked to a number of adverse health effects, including not only digestive disturbances, but also adverse systemic effects ranging from fatigue to skin problems to inflammation and aching joints. So the composition of the stool, including the microbial composition and metabolite profile, can also serve as biomarkers of effect, providing clues about gut health. For example, the US Food and Drug Administration has approved several tests for the detection of colon cancer based on the detection of inflammatory molecules on this tool. Some microbial metabolites are also being used as biomarkers of disease. For example, the microbiome products unique bile acids like litocholic acid and deoxycholic acid. So a high fecal litocholic acid and deoxycholic acid ratio is a biomarker of susceptibility for colorectal cancer, breast cancer, and gallstones. So when we look at the biomarkers associated with inhalation exposures and respiratory toxicology, as we all know, inhalation is another common route of human exposure to environmental contaminants, and as of 2021, air pollution caused over 12 million deaths per year worldwide. So this is a big problem, as you see. The respiratory system, which includes the nasal cavity, trachea, and lungs, is a primary target of inhaled environmental contaminants, but it's important to note that inhalation is not the only way in which the respiratory system is exposed to toxic chemicals in the environment. For example, ingestion of herbicide paraquat is linked to pulmonary fibrosis that can lead to respiratory failure and even death, and injected chemotherapy drugs can have serious respiratory side effects. So types of environmental exposures that cause pulmonary toxicity and biological matrices that can be used to measure biomarkers of exposure and effect in the respiratory system, as you see from this slide. So it is equally important to note that inhalation exposures do not solely impact the respiratory system. So secondary effects from inhaling air pollutants include increased risk of cardiovascular diseases, metabolic disorders, immune dysfunction, and even neurological disorders. So interestingly, inhaled substances can reach the brain directly via the olfactory nerves that connect the nose to the brain. This is an interesting point also. Exposure to airborne toxic substances is strongly associated with the respiratory distress and disease. So also air pollutants can result from anthropogenic sources such as tobacco smoke and traffic, or natural sources such as wildfires. Environmental air pollution includes particulate matter and various chemicals, including nitric oxides, volatile organic compounds, and ozone. It also includes indoor and outdoor allergens such as house dust mites, pollen, mold spores, and mycotoxins that can trigger respiratory symptoms and exacerbate conditions like asthma. Additionally, certain occupational exposures have been identified as having detrimental effects on the respiratory system. For example, inhalation of asbestos fibers or silica dust can cause lung fibrosis, scarring, and cancer. Pesticides, such as organophosphates and carbamates, can also cause respiratory systems and lead to asthma and chronic obstructive pulmonary disease. The toxic chemicals can differentially impact the respiratory system depending on the duration and intensity of exposure. Therefore, biomarkers of exposure, effect, and susceptibility are essential for monitoring and mitigating exposures to prevent diseases and improve lung health. The need for biomarkers of respiratory exposures is particularly important because it is difficult to sample the lung directly. And several different biological samples can be used to determine what is happening in the lungs following exposure, as mentioned in the last table. These include exhaled breath condensate, induced sputum, blood, and urine, all of which are minimally invasive approaches often used in combination with measures of lung function to understand onset or worsening of lung disease. The more invasive approaches, such as bronchoscopy or bronchoalveolar lavage, have more restricted use and are only obtained in certain clinical settings. The collection of EBC is increasingly used because the instrumentation for collecting and measuring EBC has become more widely available. So during EBC collection, exhaled breath passes through a cooling device resulting in the accumulation of substances in exhaled air. So concentrations of these substances, which can include environmental chemicals and biomolecules found in the cells of the respiratory system, are influenced by lung disease and thus can be used as biomarkers of not only chemical exposure, but also the pulmonary response to inhaled respiratory toxicants. Similarly, induced sputum allows for the analysis of different types of cells in the airways and is widely used for characterizing airway inflammation. The elevated presence of white blood cells, immune cells important in the body's defense against bacterial and parasitic infections, as well as allergens, is an indication of inflammatory response to environmental exposures. These cellular biomarkers can be also evaluated using more invasive techniques of bronchoscopy and bronchoalveolar lavage fluid collection. During these processes, a tube with a tiny camera on the end is inserted through the nose or mouth and into the lungs. In a bronchoscopy, a small piece of lung tissue is collected to be examined. In BAL, sterile saline is introduced into the lung and immediately suctioned to collect the saline solution for analysis. So that these are new approaches and more useful ones. Compared to the sputum analysis, which provides information about the nose and mouth, bronchoscopy and BAL allow for the sampling of deeper sections of the respiratory tract with less microbial contamination and can be useful for understanding local inflammation within the lungs. When significant respiratory toxicity has occurred or there is a widespread inflammation throughout the body following an inhalational exposure, it is common to analyze specific lung-related proteins in the blood or urine as biomarkers of these effects. An early response to environmentally induced lung injury is increased permeability of the lung epithelium. the layers of the cells that line the lung that functions in barrier protection, much like the epithelial cells that line in the gastrointestinal tract. So increased permeability allows epithelial proteins to leak into the blood and then subsequently pass into the urine. CC16, a lung cell protein, is an example of a blood and urinary biomarker of increased lung permeability. In children, urinary levels of CC16 are a validated biomarker of the development of wheezing and were found to be elevated following acute exposure to PM in wildfire smoke. So biomarkers of susceptibility have provided important clues for understanding why certain individuals may be more susceptible to respiratory problems in response to toxic exposure and how environmental exposures may contribute to the development or exacerbation of respiratory diseases. For example, specific genetic variations in the detoxification gene glutathione estrancease M1 and inflammatory cytokine gene TNF-alpha are associated with COPD, while the genetic variations in the inflammatory cytokine gene interleukin 33 and the interleukin 33 receptor gene ST2 and interleukin 1 RL1 are linked to asthma. So when we look at the biomarkers of neurotoxicity, until recently, there have been few biomarkers for assessing environmental exposures in the brain or the effects of the environmental stressors on brain function or neurological diseases. The need for brain-specific biomarkers is underscored by the increasing incidence and costs of neurological diseases such as autism, ADHD, Alzheimer's diseases, and related dementias, and the growing recognition that many, if not most, complex neurological disorders arise from interactions between environmental stressors and genes of susceptibility. As a result, there has been an upsurge of interest in identifying biomarkers for assessing the impacts of environmental contaminants on the brain. This interest has driven a number of emerging approaches involving brain-derived molecules released into the systemic circulation, in vivo imaging of the living brain, and epigenetic biomarkers in accessible peripheral tissues as proxies for epigenetic changes in the brain. Several blood-based biomarkers are now in clinical use for detecting and monitoring the progression of Alzheimer's disease and related dementias. These include the neurofilament light chain, NFL, and phosphorylated tau, abbreviated as phosphotau. NFL is a cytoskeletal protein uniquely expressed in neurons. NFL is a very stable protein that functions as part of the backbone of the neuron, providing tensile strength to support the long processes extended by neurons. Because neurofilaments are relatively resistant to the cellular processes that normally break down proteins released by dying cells, they can be transported out of the brain tissue and into the circulatory system. Tau is a cytoskeleton-associated protein that normally functions to regulate the assembly and disassembly of the cytoskeleton in neurons. Phosphotau is a biochemically modified form of tau that is involved in the formation of neurofibrillary tangles, which are a hallmark characteristics of Alzheimer's and other tauopathies that cause dementia. Accumulating levels of phosphotau in brain tissue begin to leak into the circulatory system, so that the blood levels of NFL and phosphotau are positively correlated with the severity of damage in the brain and are proving to be useful biomarkers for environmental health scientists studying the role of environmental contaminants, such as pesticides and air pollution, in promoting neurological disease. So, also extracellular vesicles are another exciting development in the field of blood-borne biomarkers of exposure and effect in the brain. So EVs are membrane-bound sacs that are shed by all cell types into the extracellular space before migrating into the circulatory system. They are found not only in the blood, but also in cerebrospinal fluid, saliva, urine, and even exhaled breath. It was originally thought that EVs were a mechanism for cells to rid themselves of waste products. However, it is not known that EVs carry bioactive molecules, such as proteins and nucleic acids, that can influence the function of recipient cells. In other words, EVs are a form of communication between cells and tissues that are physically distant from each other. EVs are important in maintaining health and promoting disease. So there is now evidence that EVs carry molecular signatures of the cells from which they originate, suggesting the possibility of distinguishing EVs in bodily fluids that were derived from the brain from EVs generated in other organ systems. It is also now recognized that toxic chemicals and diseases can change EV cargo also. Whether specific chemical exposures or specific diseases are associated with unique molecular profiles of EVs derived from the brain is not yet known, but if proven to be true, molecular profiling of brain-derived EVs found in accessible body fluids will be a powerful tool for assessing the neurological impacts of environmental contaminants. So in vivo imaging techniques have long been used by neurologists to assess biomarkers of health and disease in the living brain. In vivo imaging has been used to a more limited extent in environmental health studies, exemplified primarily by the use of X-ray for quantifying the deposition of lead in bones, a validated biomarker of lead exposure. In the brain, magnetic resonance imaging, MRI, is an established tool for monitoring exposure to manganese. So MRI is also used to monitor neuropathological changes in the brain, including a breakdown of the blood-brain barrier. New MRI techniques are being actively developed for monitoring how cells in the brain signal to one another. Brain function can also be assessed using PET imaging. Whereas MRI relies on the tracking the spin of water molecules in the brain when exposed to a magnetic field, PET imaging involves injecting a radiolabeled substance that recognizes and binds to a molecular target in the brain. So the PET scanner detects the radioactive signal, and the amount of radiolabel is a biomarker of the level of the expression of the target molecule. PET is being used to quantify oxidative stress and inflammatory responses in the brain, both of which are thought to be important mechanisms by which many environmental contaminants promote neurological diseases. PET can also be used to assess the impact of toxic chemicals on selected neurotransmitter systems, for example, dopamine, which is altered in Parkinson's disease. Epigenetics represents a third approach for assessing the impacts of environmental pollution on the brain, as seen in the figure. So in this figure, as we all see, epigenetic predictors unveil health risks to pollution exposure. In the left, in a health environment, typical epigenetic modifications are established, which reduce risk of adverse health effects, but in the right, in contrast, exposure to environmental pollution can alter epigenetic patterns, which may be used to predict future health risks, such as cancer or neurological disorders. So the term epigenetics refers to heritable modifications in our DNA that alter gene expression without changing the gene sequence. So these modifications act as switches that determine when and how a gene functions to coordinate the expression of functionally linked genes at appropriate times during development. These epigenetic modifications are regulated by environmental cues both within and outside the organism, allowing the genome to respond appropriately to changes in its internal and external environment. In summary, epigenetics modifies the activity of DNA, which is critical for enabling genetically identical cells to differentiate into morphologically and functionally distinct cell types, such as neurons and muscle cells, and to respond to dynamic environmental influences. The epigenome, a term that refers to the complete set of epigenetic changes in an individual, can be modified by diverse environmental stressors, including pollution, malnutrition, and maternal stress. Because epigenetic modifications are necessary for a normal development, aberrant changes in the epigenome as a consequence of exposure to environmental stressors can alter gene expression to increase the risk of disease. Environmentally induced epigenetic changes may also serve as biomarkers of environmental exposures, and because epigenetic marks can be stable, these may provide clues about previous exposures as early as preconception. Medical tests are currently being developed that use epigenetic marks from DNA in blood samples to screen patients for cancer risks. These capabilities are particularly helpful for screening individuals who are at high risk of occupational-induced cancers, such as firefighters, military personnel, agricultural workers, and many others. Having access to early intervention screening using epigenetic biomarkers can be life-saving. Experimental animal studies have demonstrated that the neurotoxic chemicals can alter the epigenome in the brain and that these changes are linked to changes in gene expression in the brain that are associated with neurological disease. Such observations suggest the exciting possibility of using epigenetic biomarkers to evaluate the impact of pollution on neurological health. However, assessing epigenetic biomarkers relevant to the brain represents a significant challenge because it is typically not possible to sample the brain directly. However, human placental tissue, which is usually discarded as medical waste at birth, is also accessible. Experimental animal studies have shown that the prenatal exposure to a mixture of polychlorinated biphenyls (PCBs) detected in the blood of pregnant women at high risk for having a child with a neurodevelopmental disorder resulted in shared epigenetic patterns in the placenta and the brain of the offspring that were associated with developmental neurotoxicity. These studies not only provided insights into the complex interplay between PCB exposure and neurodevelopmental disorders, but also highlighted the potential for early identification of exposures associated with increased risk of neurological disease using epigenetic biomarkers in the placenta. Although epigenetic biomarkers can be useful for informing human health, but there are some limitations. First, epigenetic patterns vary between cell types. For example, muscle versus neuron. Therefore, it is important to consider how epigenetic variation in one tissue source can be used to infer health effects elsewhere in the body. Second, epigenetic modifications are dynamic, meaning they can change throughout the lifespan of an individual. Thus, epigenetic patterns present at birth are likely to be much different in the aged individual. Nonetheless, epigenetics offer another tool for assessing environmental impacts on the brain as well as the rest of the body. So when I want to conclude my talk, it has become increasingly apparent that individual risk for many complex diseases is determined by not only the individual's genetic substrate, but also the individual's exposure to environmental stressors. Environmental stressors known to confer disease risk include lifestyle, such as diet, physical activity, sleep patterns, mental stress, etc., as well as the environmental pollution. Medical and public health strategies targeting lifestyle factors require significant personal motivation and have been limited success at the population level. In contrast, environmental pollution represents a class of risk factors that can be changed by public policy, and it has been argued that targeting positive environmental exposures will have a broader and more significant impact on reducing diseases burden than modifying proximal individual level risk factors. And reducing environmental pollution is clearly feasible, as evidenced by the success of lead abatement policies around the world. Such changes will require compelling evidence causally linking specific environmental pollutions to increased incidence and/or earlier onset of the disease in order to motivate policy and lawmakers to change standards. There is also a pressing need for research to identify pollutant sources that can be controlled by regulation. Generating this type of evidence will require the use of validated biomarkers of exposure, effect, and susceptibility to demonstrate strong associations between specific environmental pollutants and specific diseases, determine safe exposure levels, and identify susceptible subpopulations. So thank you for your attention. I think I obey my time. I don't exceed my time. So thank you for listening to me. I hope you are not bored.
Thank you, Prof. Iker. What great informations and great presentations. Thank you very much for sharing it with us, Professor Iker. I'm sure it's very useful, especially here for all participants and also our young scientists here. I would like to sum up the main points here, the presentation of the use of biomarker in deciphering human health impacts on environmental contaminants, provide an insightful overview of how that biomarkers serve as critical tools in understanding the complex relationship between the environmental exposures and human health outcomes. Our guest speakers highlighted three main categories of biomarkers, like the exposure biomarkers and effect biomarker and susceptibility biomarkers, explaining their role in detecting and quantifying the presence of contaminants, early biological effects, and also the individual vulnerability, respectively. Several case studies illustrate that the practical application of biomarkers in monitoring pollutants exposures, also in the predicting disease risk, emphasizing their potential to improve environmental health risk assessments and guide preventive interventions. Overall, that the presentation underscores the growing importance of biomarkers in environmental health issues and also the health sciences, and call for continued interdisciplinary collaboration, I think, in the near future to refine these tools for clinical and regulatory use. Thank you, Professor Iker. I think we open for Q&A sessions for these events. I think we have one questions from our audiences here. Thanks for your presentations. It's from Miss Musfira. It was an interesting presentation. She has two questions, Prof. Iker. Here's first, could you explain how to overcome the methodological challenges and implement best practice for standardizing biomarker measurements for environmental exposure assessments across diverse populations? That's for the first question. And the second one, that she asked about how does PET directly affect the development of Parkinson's disease? Could you got the points, Professor Iker?
Can you repeat the first question?
Yes. First question, I think it's in the chat box. I think it's from Iker's. First question is, she asked about, could you explain how to overcome the methodological challenges and implement the best practice for standardizing biomarker measurements for environmental exposure assessments across diverse populations? I think kind of the methodological challenges, Prof. for standardizing biomarkers measurements for cross populations?
Yes, there are some researches about these biomarkers, so that there is a big problem, maybe the cross populations. So they are working in these to get rid of these problems. So I think that it will be a great opportunity to find new techniques or methodologies to see the differences of the biomarkers in the cross populations.
Yes. Bu Muspira, do you got the point, right, for the first of your questions?
Okay, thank you for
Yes.
your answer.
So for the second one, that she asked about how does PET directly affect the development of Parkinson's disease, Prof?
The PET imaging is a newly technique that is used for some neurological diseases such as Alzheimer's or the dementia-related dementia, so that it is going to be developed. Maybe some new additive techniques can be related with this PET technique, so that imaging will be more qualified and more useful technique, I think.
Oh, okay. Thank you so much. I think for the first questions, because Ms. Muspira asking about the diverse populations for the biomarkers, I think, Prof. You also mentioned it on your presentation. You mentioned it about we can say that this SNPs like single nucleotide polymorphisms or variability between the cross population. I think it's this kind of also genomic variants that we call the SNP is also can be implemented for diverse population, right?
Yes, you are right. The single nucleotide polymorphisms are making diversity in the population so that we can go on with these polymorphisms.
Yes, yes. Okay, thank you so much for Professor Ikaratas. I think so we also waiting for another questions, and before we got question from others audience, I think I would like to ask also Professor Ikaratas, because your presentation is really challenging, and I would like to ask about the challenges of the analytical or clinical validations of biomarker for regulatory and clinical applications like the assay accuracy and also the precision and reproducibility. Do you got this kind of the issue about the challenges of the analytical and clinical validation?
Sorry, I am. Am I in the Zoom? I'm not seeing the.
Stuck. I think stuck from Turkey, right? Hello, Prof. Iker. Okay. Okay, I think Professor Iker Ates, because I think yes, we are waiting for him to entering our Zoom meetings, and I think before we are going to interact with the Professor Iker Ates, also we are waiting for another questions from our audiences. Is there any questions? You can chat. You can writing it out on our chat box, or you can directly when our guest speakers join the meeting again.
Hi again. Do you see
Okay.
me?
Yes, Professor Ikers, we see you
Sorry,
again. Okay.
I lost connection. Sorry.
Okay, no problems. We can continue.
Okay.
Yes, Professor Iker. Do you got my questions?
Can you repeat again?
Yes.
I lost connection.
My question is about the challenges in the analytical or clinical validations of biomarker, Prof. like the issue of the accuracy and also the precision and also the reproducibility.
Yes, there are some validations about the reproducibility and the other corrections or the other problems, so that there will be need of some alterations or some works to make these more correct or more reproducible or more feasible. So that
Prof.
we need studies.
Prof. Iker, could you open your camera?
Now my camera is open. Do you see me?
Not yet.
Is there a problem again? So let's.
Yes. Hello, Professor. Okay.
Hello again. I now see you.
Okay. Hello.
Hello.
Yes. Could you open your camera, Professor?
My camera is open.
Yes. Okay. Yeah.
Now,
now we see you, and then could we continue it?
Yes, please.
Yes, Prof. My question is about the. I asked about the analytical and also the clinical validation, Prof. About the biomarkers issue about because some of the issue about biomarkers about the accuracy and also the precision and also the reproducibility, right? What do you think about this kind of the issue?
Yes, the analytical validations are needed to make the works correct, so that the need of the more modifications may be done to more make it more accurate or more feasible or more reliable.
Okay. Okay, we got points. I think we have another questions from our audience, Professor Iker, from Mr. Ridwan Ansari. That was interesting topic, Professor Iker. I just wondering how long the biomarkers of gut microbiota profile might reflect the exposure of poor dietary patterns of or food toxics. That's for the first questions. And for the second questions, how about the risk of potential bias for measurement gut microbiota from the feces? How we control it? He has two questions. Yes.
The microbiome is very important, as nowadays it gets importance for measuring the foodborne problems, because the gut is the second brain, as you all know, and maybe it has more importance in getting on the surface, so that the gut microbiome makes the biomarkers, make it the biomarkers to see that what is happening in the gut. So the differences or the changes in the microbiome shows us the problems, the toxic effects of metals or other foodborne illnesses or foodborne contaminants. So that it is important, it has importance, and it is nowadays making more and more getting importance. So the second is the bias, I think, the second question.
Yes. Second question is how about risk of potential bias for measurements gut microbiota from feces?
Yes, there may be some bias in measuring from the microbiome. Maybe the problem should be about the potential of the microbiome or potential of the gut. Maybe the differences or the differences between the individuals can be the bias for this reason, I think.
Okay, thank you. Mr. Ridwans, do you got your answer right?
Yes, thank you.
Okay, thank you, Pak Ridwan. And thank you, Pak Ridwan, and also the other question. I think the last questions, Professor Ikers, I'm still curious about how do regulatory agencies like the FDA or EMA approach for qualification and approval biomarker, because we are the scientists, right? And what are the critical steps researcher must follow to achieve regulatory acceptance?
The regulatory agencies such as you mentioned, EMA or EPA, I mentioned from the US agencies, they are working on the biomarkers so that they are getting more works to find new biomarkers or new introducing things to us researchers to find and to get knowledge about the biomarkers, new biomarkers or new techniques in the science. So that, as you know, biomarkers is a big ocean, so that we are going to find new ones, maybe, and the agencies have the major role to find these new biomarkers or new insights in these topic.
Okay. Okay, Professor Iker Atas, thank you very much. And I think I have a lot of questions because we have limited time, Professor Iker Atas, so that we can, I think, we can discuss it later on about this kind of the issues, Professor. And the Professor Iker Atas, on behalf of our organizing committee, I would like to present the certificates for you for your insightful presentations and the valuable contribution to our event this morning.
I all thank you for this nice conference. I thank you, Dr. Ihram, to invite me in such a wise symposium. Thank you for all organizing committee, and I thank you for all the audience for listening me patiently. I hope I answered all the questions, and thank you again so much.
Okay, thank you very much, Professor Iker Atas, and see you again at Ankara University at Turkey. Thank you once again, and because of the limited time, we really appreciate the enthusiasm for all our participants about this event for this morning. And for the next, we would like to hand over again to our MC, Ibu Putri. Thank you so much.
Okay, thank you very much. Well, it's really, actually, this is amazing presentation from our keynote speakers. Thank you for Associate Professor Ilker Ates from Ankara University of Turkey as a speaker, and also the moderator, Mr. Lalu. Thank you very much. And thank you for everyone for being the part of the special moment for this day in Ahmad Dahlan International Conference on Health Sciences 2025. Actually, the next agenda is the poster presentation session, but actually, before that, let me to close this special occasion, special moment of this day, because there are a lot of things that we can get today, and I would like to express my gratitude to all of you for our esteemed guests, and also for participating in this event and also making it huge success. It's really wonderful. Your contributions and presence have added a great value to this conference actually, and we hope that you found it insightful and also educational, and have a great day. See you again on the next day and the next year. Wassalamualaikum warahmatullahi wabarakatuh.